Good morning. I am Sachin presenting the seminar for a pre-PhD coursework, which was organized by Department of Chemistry and HRDC, Dr. Babasaheb Ambedkar Marathwada University, Chhatrapati Sambhajinagar. My research topic is a search for a new antimicrobial agent having nitrogen and sulfur containing bioactive heterocycles. I will doing PhD under the able guidance of Dr. A.W. Suryanshi sir, Professor, Department of Chemistry, Sri Madhavrao Patil, Mahavidyalaya, Murum. This is outline of my presentation, which includes introduction, literature, way, significance of our objective, importance of 1,2,3 triazoles, synthesis of 1,2,3 triazoles, future plan of work, expected outcome, and references. First of all, I would explain introduction. Heterocyclic compounds are nothing but cyclic organic compound which consist of one or more heteroatom in their structures. The common heteroatoms are oxygen, nitrogen, and sulfur. The heterocyclic compounds are classified into two classes: aromatic heterocycles and aliphatic heterocycles. Some examples of aromatic heterocycles are given as pyridine, pyrimidine, quinoline, isoquinoline, pyrazole, tetrazole, triazole, pentazole, indole, thiophene. Whereas aliphatic heterocycle consists of oxirane, oxetane, azitidine, tetrahydrofuran, tetrahydropyran, dioxane, dithiene, pyridine, morpholine, thymorpholine, and many more. Heterocyclic compounds play vital role in various fields such as medicinal, pharmaceutical, organic chemistry, industrial chemistry, polymer, polymer chemistry, biochemistry, and also in food industry as well as in agricultural field and many more areas of the sciences. These compounds are present in vitamins, biomolecules, drugs, and biologically active compounds. These compounds have certain therapeutic activities, and these therapeutic activities consist of antimicrobial, antifungal, antitubercular, anti-diabetic, anti-cancer, anti-inflammatory, anti-HIV, anti-malarial, antidepressant, anti-tumor, antiviral, and insecticidal, etc. 90% of the newly synthesized drugs consist of aromatic heterocycles, and which will contribute in the field of the medicinal chemistry. Some of the top 200 banded representative drugs consist of aliphatic heterocycles in their fragment or in their structures are given in figure 1. These examples of pascanazole, ciprofloxacin, penicillin, phenytoin, terazosin, etc. Similarly, some top 200 branded representative drugs having aromatic heterocycles structures in their molecular framework are given in figure 2, and which consist of omeprazole, fluconazole, isoniazid, tizanidine, celecoxib, allopurinol, etc. These are the figure 2 drugs. Keeping the importance or seriousness of these nitrogen and sulfur containing bioactive heterocycles, my PhD work will be undertaken on the topic a search for new antimicrobial agents having nitrogen and sulfur containing bioactive heterocycles. Next, literature survey. Literature survey reveals that nitrogen and sulfur containing bioactive heterocycles are more important and they have antimicrobial activity. And antimicrobial activity consists of antifungal and antibacterial activity. Some of the repeated literature survey are given as Chennakeswara Rao Kela, and it has synthesized a novel class of 1,4-disubstituted 1,2,3 triazoles in good yield by copper plus azide alkane click reaction method. Most of the compounds exhibited promising activity against gram-positive and gram-negative bacteria. Zebula 8L have been synthesized thiazoles and oxidazoles by multi-step synthesis, and most of the compounds have highest antimicrobial activity as compared to the standard drug streptomycin. Pengeo and co-workers synthesized a series of tetrazole pyridine hybrid compounds and screened for their in vitro microbial and anti-inflammatory activity. All the synthesized compounds shows promising activity, that is antimicrobial activity against bacteria like S. aureus, E. coli, B. subtilis, Epimegatus. Next, a new class of methylthioline pyrimidine 1,3,4 oxidazole, 1,3,4 thiazole, 1,2,3 thiazoles were synthesized by Madhushekar and co-workers. These compounds are tested for antimicrobial activity and shows promising antimicrobial activity against the bacteria. A new class of pyrazole containing S-triazine derivative were synthesized by Sharma et al. Synthesized compounds shown antimicrobial activity against respective bacterial strains. Most of the most of them shown potent microbial activity. Tri-substituted thiazoles prepared and studied by good molecular identity for their antimicrobial activity as well as their theoretical calculation. Then significance of the work. The research will provide a library of new nitrogen and sulfur containing heterocycles. Research will also existing the current antimicrobial agents. The research will also helpful to improve the various scope and limitations of market available drugs. The screening of antimicrobial activity may give the library of potential therapeutic agent, which will surely contribute in the field of medicine and pharmaceuticals. And this research is important. This research will be important for the guidance in the future research. Objective of the work. Following will be the main objective of the proposed research problem: to design a library of new nitrogen and sulfur containing heterocycles, to use excellent green reaction conditions for synthesis of new nitrogen and sulfur containing heterocycles. All the synthesized compounds will be characterized by spectroscopic technique like IR, H1NMR, C13, HRMS. All the compounds were screened for their antimicrobial activity. The importance of 1,2,3 triazoles. These 1,2,3 triazoles are transbioesters of amide bond. This transamide bond, and this is 1,4 disubstitute 1,2,3 triazoles. These are similar, and this cisamide bond and 1,5 disubstituted 1,2,3 triazoles are similar to cisamide bond. These are the similarity between the 1,2,3 triazoles and amide bond. These 1,2,3 triazoles are most important because they are easily and reduce-selectively synthesized by proper first accelerated reaction by click chemistry. Most of the compounds are stable at acidic and basic condition. These compounds are stable in oxidation reduction reactions. And most important thing about these compounds, uh, all these 1,2,3 triazoles binds to many biological target with high affinity due to presence of nitrogen atom in the triazole ring. These compounds have promising drug core structures and they are highly bioactive. Keeping this view or importance of 1,2,3 triazoles, I have been synthesized 1,2,3 triazoles. This is a scheme of which can be carried out by me. In first step, Substituted salicylaldehyde and propyl bromide will be reacted in presence of K2CO3 and DMF as a solvent to form the substituted salicylaldehyde alkyne. This alkyne and substituted different substituted azides on cycloaddition reaction in presence of copper sulfate, sodium ascorbate, and PEG as a solvent to form 1,4-disubstituted 1,2,3 triazoles. Synthesized compounds are shown as below. 12 compounds have been synthesized by me. This one is a synthesis from plain triazole, 2-methyl triazole, 3-methyl triazole, 2,3-dimethyl, again 2,4,6 trimethyl, 2-methoxy, 4-methoxy, 4-bromo, 2-chloro, 3-chloro, 4-chloro, and 4-chloro. The synthesized compounds are evaluated for their antimicrobial activity, or antimicrobial assay was performed. The pathogenic bacterial cultures were exposed to for their compounds. All newly synthesized compounds were screened for their antimicrobial activity and antifungal activity against the different strains. For antimicrobial activity, Streptococcus aureus ATCC 6538, Bacillus megaterium ATCC 2326, Bacillus subtilis ATCC 6633, gram positive, and E. coli ATCC 8739, type P ATCC 9207, and Aureus ATCC 13048, and Pseudomonas aeruginosa ATCC 9027. Gram-negative pathogens were used. For evaluating antifungal activity, Aspergillus niger ATCC 16400, Candida albicans ATCC 10231 fungal pathogens were used in this study. Some of the compounds found to display potent antimicrobial activity against pathogenic antibacterial and antifungal strains as compared to the standard reference drug tetracycline. Future plan and work. Synthesized compounds will be purified and using ethanol solvent. Determination and yield of physical constant of all newly synthesized compounds. Characterization of all compounds can be carried out by using various spectroscopic techniques like H1NMR, IR, C13, and HRLS. Design and synthesis of new nitrogen and sulfur containing bioactive heterocycles. Then expected outcome. The expected outcome of the proposed research will be surely contribute in the field of research and medicine, which will expand the current state of knowledge. The proposed research may develop the library of new potential bioactive antimicrobial agent, which will surely contribute in the field of medicine and pharmaceutical. The other expected outcome will be in the form of paper and poster presentation in the national and international conferences, reviews and research article in the various journals and internationally reputed, as well as national and international patents. These are the references. I will thankful to my guide, mentor, Dr. A.W. Suryansh sir, Professor, Department of Chemistry, Sri Madhara Patil Mahavidyalaya Murum, Taluka Maradi, Dharamshi. I will also thankful to pre-PhD coursework organizing committee, which consists of Dr. Bonle madam, Dr. Chaya Sonara madam, and Dr. Sachin Deshmukh, which is central coordinator. Again, I will thankful to all of you. Thank you.